Hi, Katarina.
Hi, Victoria. How are you?
I'm well,
Nice
thank
hearing
you.
you.
Yeah, nice to hear you too.
How is everything?
Everything is great. I'm outside right now on the porch at the beach, and it's beautiful and cold and windy, and watching the sunset.
Oh, beautiful. Are you in California?
I
Or
am, yes.
nice.
Mm.
That's nice. I just set up the
Oh,
presentation.
the room looks fabulous. You made it look great.
Thank
Oh,
you.
I was just reading the paper too.
That's really interesting.
Mm-hmm.
Okay. Paper shared. Good. All good. Yeah. This month is really busy. Next month I still don't have that much planned. I have to catch up because I didn't do much last month, you know,
Hmm.
on vacation. So
Yeah,
let's
yeah.
see.
Oh, it'll fill. It will.
Yeah, well, since we planned this like 2 months ahead around, so you know, there's a delay in spots that are kind of, but we'll have a few rooms. It's fine.
Welcome, friends who are arriving.
Hey everyone, feel free to join us on the stage to ask questions, participate. Hope everyone's doing well in the new year.
Oh, shoot. I was reading what I wanted to read. It's actually in the top. Never mind. My question's answered. It's immune system that I didn't read. I seen the molecular disabling switch, and you perked my ears up big time. But immune system kind of defines it a little further that I wasn't exactly looking for.
Well, maybe you'll be surprised and interested.
For sure, maybe.
Yeah, great. All right. Well, enjoy the talk. Thanks for coming.
Well, it's really important, right? There's a lot of autoimmune disease, and other, you know, diabetes is also falls under the category of autoimmune diseases. Even in cancer, it's important to know that switch, because cancer actively sometimes turns it off. So it has really a broad range of importance for health. So it will be really interesting.
I was looking. I didn't see reference in the two articles I'd read to autoimmune applications, and that's what I was. I mean, everything is so important, but I'm so curious about if that's a part of this work as well, or ideas for the
Well,
future anyway.
yeah, this work is to figure out how it works, like the the immune system, molecular system, but later on it will have implications, I guess, in those fields. So yeah, it was for sure hard enough to to find that switch. I mean, it's really great work.
Yeah, it's gonna be great.
Yeah, welcome everyone. Thanks for coming. Feel free to share the room. Our guest speaker will be here soon. We'll start on top of the hour. In the meantime, feel free to check out the slides, the paper. Let me also share like a general public press release. Didn't I have it? A link. So sometimes it's easier for people to understand what like the context is and the significance.
Oh, welcome, Hamish. So happy to see you.
Yeah, welcome. Can you hear us? The unmute button, like the button to unmute, is all the way on the bottom right. There should be a little mic. Yep.
Oh,
There you
yeah.
go.
Oh, good. Cool. That's all right. Sorry, I didn't realize that button.
How are
Um,
you? Everything good?
I'm good. Yeah, I'm good, thanks. Yeah. How's the connection? Sounds all right. Does it?
Yeah, I can hear you well. Can you hear us well?
Yeah, that I can hear fine. Yeah. Yeah. No worries. Yeah. No, I'm good, thanks. It's yeah, it's different. So it's 1 p.m. here almost.
Yeah, and you have nice weather. No?
Yeah. Exactly. What's it like where are you? You're in New York, are you?
Yeah, but it's a relative for New York. It's a very mild winter so far. No
Oh, that's
snow.
good.
Not really. There were a few days where it got really cold, where stuff was freezing, but so far not too
Yeah.
much. Which is
Yeah,
good,
right.
of course, bad in the bigger picture. I mean, Europe, same thing. My parents live in Germany and Portugal, and yeah, very
Yeah.
mild in Germany too. Almost no
Yeah,
snow.
right. Yeah, we had a really long winter. Like our winter's pretty mild, but it was like it seemed to go on for like way longer than normal. And but summer finally came and it's been really good. Like we've had like 30, 35 degrees for like about a week and now it's gone kind of cold again, but that's that's our weather's pretty unpredictable.
Yeah, I think in Portugal, the summers are not, at least in the north, the summers are not really that warm anymore. Um, like, I don't know. So it's
Yeah.
been kind of weird, but
Yeah, yeah,
well,
definitely.
it was to be expected, not surprising.
So I realized I didn't send you like a bio or anything, but sorry about that.
Oh,
A
don't
bit
worry.
late.
I'll check. I checked you out. No, I just went on the lab website and on LinkedIn, so it's all good.
Yeah, no worries. Yeah, yeah, no worries. It's probably
It
yeah,
sounds
that's probably what I would
creepy,
send you.
but
That's all right. No, no worries. Don't worry. I think I did it to you as well, so don't worry.
good. Okay. Yeah, welcome everyone. Hi, Katie. You want to come up? Um, yeah, welcome everyone. We'll start in a few minutes. Feel free to, in the meantime, check out the paper, the article, and the presentation. And welcome, Katie. Meet Hamish.
Hello, how are you?
Good, thanks. Your accent sounds familiar.
As does yours. Welcome. I'm really excited for your presentation.
Oh, great. Thank you. Yeah, thanks for having me.
How has your start of the year been, Katie? We haven't spoken in a few weeks, I think. So hope all is well with you.
Yeah, everything's good. And how about yourself? It feels like too late to say Happy New Year, but Happy New Year since we haven't spoken. But I'm excited for these rooms to be back and chatting with you all and nerding out on science.
Yeah, thanks. Happy New Year to you too. And we'll have the Chinese New Year coming up. So it's this weekend, right? If I'm not mistaken. So we can say Happy New Year. Okay.
It's always great to wish happy anything at any time, you know? I don't think there's too much of that.
This is true. This is true. And hi,
Yeah.
Victoria.
Yeah, hello, Katie. Happy New Year. It's great
Happy New
to have
Year.
you back. Just like the olden days.
I know, I
Science people
know.
together again. It's good. It's going to be a good room. We're looking forward to this, Hamish.
Oh, good. I'm glad. Yeah. Oh, yeah. So, would you like me to get started, or are you sort of waiting
Oh yeah, you can relax
for people?
for 2 more minutes.
All right.
Yeah,
Sounds
we
good.
we start at the hour. Well, you're good. You can just relax and we'll let you know.
Okay. No worries. So, where's Katie? Whereabouts are you situated, and Victoria?
I am currently in like northern New South Wales, but I've been
Oh, cool.
living in South Africa, gosh, for like 12 years.
Ah, right. Okay. Yeah.
Yeah, and I
Yeah,
was at
nice.
the genetics department at Stellenbosch University, so I'm really excited for your presentation.
Oh, awesome. Yeah. Yeah, nice. Cool. And Victoria, whereabouts are you?
Right now I'm in Southern California and it's just beautiful outside. We had pretty exciting rains and flooding and like rivers overflowing their banks and train trestles getting washed away and that. But it's calm today. It's just been so windy, and now today it's really beautiful.
Oh, nice.
And I
That's
just
great.
enjoyed a fantastic sunset with just so orange, and it's great.
That's amazing.
Well, I'm glad we got to all have a time to chat and meet. And I think we can start. And hi, Nikita. Welcome. I haven't seen you in a while, too. I hope all is well.
Hello, hello after a long time. One of my favorite clubs every time.
Thank you.
Being
Okay, we can get started then. Welcome everyone to Science Society, and of course, a special welcome to you, Hamish. And before we start, let me give like the audience a short introduction so they can get to know you a little bit. So, uh, Hamish, he's a senior research fellow at the University of Melbourne in Victoria, Australia. And he went to a Christian college um, and then later did his bachelor with honors in biotechnology at Deakin University, and then his PhD at Monash University in parasitology, immunology, vaccine development, biochemistry. And he, as I said, is a senior research fellow in the Department of Microbiology and Immunology at the Dorsey Institute. And he leads a team in the lab of Professor, I hope I'm saying it right, José Tangush, where
Yeah.
he investigates how the immune system detects pathogens by a protein called MR1. And Hamish did really advances in understanding how MR1 operates on the molecular level, and he has been recognized and supported by several grants and prestigious awards. And it's such a pleasure having you here today. And we usually start off with interview that is led by Victoria. So, Victoria, the stage is yours. Thank you.
All
Cool.
right, thank you,
Thank you.
Katarina. And Hamish, you see, even though you didn't send the bio, she has her ways.
Yes.
So,
Even my
yeah.
high school you found somehow.
Right? Yeah, she has her ways. Well done, Katarina. So now we're even more looking forward to hearing your research. And we'd also like to hear a little bit about you. So if you can share with us maybe where your passion came from, and by that I mean, where do you feel that your first inclination towards science, or maybe just affinity towards science, surfaced in your life, even as a child or
Yeah, sure. Yeah. So I I always loved just finding out how things worked and all that. I think it's probably something common to scientists. And I remember a few memories. Like I was really young, maybe I was like 4 or 5. Actually, my family lived in the US for a few years, and I was there, and I remember going to a museum, and there was like all these animals, and we had these science classes there. And that was just like really, I just loved it. Every little bit of information, and I got a book on evolution and looking at the pictures of all the animals that were from for so long ago when the animals first left the oceans and things like that. And I've still got that book today. And yeah, I think just when I got through school, I just loved learning about things, and I thought I wanted to be a doctor for a long time. So it was always about like a medical doctor, because I was always interested in how the body worked. But then as I got through high school, it was just more actually about biology, and so I left high school and went to university and just did basically a straight science degree. That was that's how the way it works in Australia. And we sort of majored in biotechnology, but it was mostly cell biology and biochemistry. And yeah, I think I got to the end of university and realized that everything started to synthesize. Like, you learn about all these different parts, like biochemistry, cell biology, genetics, and you start to get a bit of a picture. Like, it starts to fit together, and I just love that. And then I started research, and yeah, fell in love with research. So that's probably a bit of a history of my love for science.
Thank you so much. That was really that was very smooth storytelling. I feel like you took us along this whole pathway, and it was really great to hear about your early memories of going to the museum. And it's it's just very encouraging to always share things that are important with youth. Like share science, share what you love,
Yeah.
is what I'm trying to say. I'm here visiting family now in Southern California, and we're right near. I don't know if anybody knows what the tar pits, the LA tar pits are. It's
Oh
it's
yes, I went there. Yeah.
okay. I was wondering where you were when you were describing these museums.
Yeah.
Yeah, and so you see that. Yeah, so you've seen that.
Yeah, I did, but I went there on a trip recently. Oh, it was like about 5 years ago. I was in the US, and I went there. That's near the big art gallery, isn't it?
Adjacent,
That
completely
right?
right there. Yeah. That's where I grew
Yeah,
up. And so to see the fossils coming out and to actually be walking, you can reach down and touch the tar that the woolly mammoths and the saber-toothed tigers were just they were drowned in there. But you can see it, and
yeah,
so it's amazing that you can think I'm walking in the footsteps of these creatures, and it just kind
yeah.
of sticks with you, like yours did. And so, yeah, thank you for also explaining, that things synthesized after you built on your studies, and it's kind of like you had this science vocabulary. And then can you please, you said you started research. How did you, from your beginning of your research, how did you come to the research that you will present to us today?
Yeah, so yeah, that's been quite a journey as well. And in science, I think it's like most of us, you can't really plan where things go. It's sort of just you fall into opportunities usually, or you take opportunities. And so I did my honors degree, which is it's in Australia. It's very different from the US, but it's a 1 year research focused end of the degree. And so I did that thesis on this parasitic disease of salmon. That's when salmon are in aquaculture, when they're farmed, they often get stressed and their immune system goes down. They get this disease on their gills. Anyway, I was doing research on that, and
Oh.
then yeah, and then I ended up just sort of working on parasites for a number of years. And I went to Edinburgh, the UK, before I did a PhD, um, and worked on a horse parasite. So I jumped around, and then I did my PhD on schistosomiasis, which is also known as the blood fluke. It's one of the most prevalent diseases in the world, really. It causes chronic illness in people in developing countries, and that's a fascinating parasite. The life cycle is just crazy. It goes from snails to mammals, and I actually worked on the Asian schistosomiasis, and that's in China. Although it is reducing, I think, as conditions improve in the rural parts. But so I was there collecting samples from buffaloes and collecting snails in the field, and that was a very exciting time. And then at the end of that, I think I always just was very interested in the fundamentals, and how do molecules work in order to dictate what you see happening on the larger scale. Um, and so I was looking for a postdoc position, and I was staying local, so I did my PhD in Melbourne, Australia, and I wanted to stay there due to family reasons. And there's a lot of really good immunology in Melbourne. And I mentioned before that I was interested in the medical side of science, so I thought I'd see how it went, trying to get a postdoc in immunology. And I went to work for Professor Jose Villadangos, who's I'm still in his lab now, as 9 years later. And so I was sort of lucky to get, he advertised the project and a position, and it just really struck a chord with me because it was all about this protein called MR1, which I'll explain to you today. And I was lucky enough to get that job. So it was a bit of a sideways step into something new, but I used a lot of the skills that I'd learned from parasitology, like the basic lab skills and biochemistry skills. Yeah, so I started working and then fell in love with a new area, which was this protein called MR1. So yeah, and
Thank you. Is that yeah? Sorry, was I cutting you off, or that
no,
was?
that's about
We're
it. Yeah,
here. Yeah, because we're here with MR1 today, right? Yeah. So your pattern of your kind of a pattern of inquiry brings us here, and it's interesting to hear about that path, because as we listen to your research, then maybe we can think of, also reasons why you might have gone in different directions, considering where you've been. So at this point, I will pass the mic completely over to you, and you can dive into your discussion, and then know that we are all here at your service to help with questions that people may put in the chat, or also friends on stage will have for you. And I want to welcome Dr. Shaw. It's so nice to see you, and welcome Eli also to the stage. And
yeah,
so, Hamish, yeah, the mic is yours. We're also happy and welcoming to you.
yeah, great. All right, thanks so much. Okay, so I'll get started, and if anyone wants to ask questions on the way, I don't mind doing that. I'm not sure if that's the general principle, but sorry, my little 3-year-old's coming to ask me a question. Do you mind just? I'm just giving a talk at this.
No worries.
Awesome. Do you mind showing Mama?
Take your time.
I want to show Mama.
Oh, good. I'm just talking to some people on the internet.
I'm here.
Okay, perfect. Thanks. All right, I'll see you soon, okay?
Okay.
Okay, sorry. Yeah, I've got a little boy called Atlas. I'm actually, maybe I should start with this, letting you know that I'm actually at the moment the stay-home dad for a few months, because my wife had a baby in May, and so now I'm looking after both. So it's a real privilege. Actually, Australia has very fortunate. A lot of my university has really good conditions for parental leave. So also for fathers to take it. So my wife's been able to return to work. So I'm going to return to the lab in February.
We
So
absolutely love that.
yeah,
And no worries if your
very,
child interrupts. I know what it's like.
yeah,
And many of us multitasking parents do, so don't worry.
yeah,
It's actually
exactly.
a really good example, of real life. So please.
Yeah, that's right.
Take
Yeah,
it
you're
off,
right.
Atlas. Atlas is welcome here.
And
Good.
the fact that like a wealthy country cannot have its economy collapse if they offer such benefits.
Yes, equal
Yeah,
paternity leave.
yeah, you're right. Yeah, I know. And I mean, yeah, it's one of those things that I sort of felt like it was almost indulgent for me to do it for a while, but then I realized that the situation, that equal opportunities is never going to get better if it's just the mothers that have to stay home and don't get the opportunity to follow their dreams as well. So anyway, it's very, I feel very fortunate to be able to do it because I spend time with my kids, but I do also miss the science, so I do work a bit in the evenings. I've got some students and keep in touch with them and all that. But anyway, we'll
No, I don't want to.
Anyway, we'll move on. We'll get into the. Oh no. I'm just all right.
You could
He's a
also,
3-year-old.
if you need to press your mute button for a second to have a little privacy, you can do that and come back whenever you
Yeah.
can.
All right, I'm going to mute for
No, no
1 minute.
worries.
I'll be back.
Yeah, yeah.
All right.
Take care of what you need. We're good. We're happy. We'll talk amongst ourselves.
Yeah, hi, it's Katie, and just maybe it's a good opportunity to say to anyone that's listening in the audience as well. I know that we've got some new people in the room. Please click on the presentation that's at the top, and you can follow along with us as Hamish goes through. Katerina also has put in the chat links to the scientific studies as well. If anyone has questions along the way, as Hamish said, he's happy to answer them. So you can pop them in the chat or feel free to raise your hand. There'll be time for questions and answers at the end, and we'll make sure that we get to you all. But yeah, again, like really love the multitasking life. I'm sure many of you that know me when I'm with my girls too, you can hear them running around in the background, and yeah, amazing chat about maternity leave and working parents life, because that's what many of us do these days.
Yeah, I wanted to also add there was a study in Portugal since they started that men can also take parental leave. It improved also the health of the relationship of the parents. That's what showed the study, since both know how it is to like participate as equal partners and educate, like being there for the children and work. So apparently it's also good for the parents. So anyways, Hamish is back.
I'm back. All right, thanks.
Welcome.
Yeah, thanks. I don't want to make it all this about about that, but it's a very interesting and important discussion, isn't it? I mean, but anyway, my 3-year-old wanted to hang out with me, which is lovely, but he's gone with his mum now, so all good. Okay, so I'll get started, and so essentially what I'm going to talk. I know that you have very diverse speakers and audience as well, and so what we work on, I'm going to try and start with a broader description, but then in order to explain the paper, I'm going to go into the nuts and bolts of the mechanics of what's happening in the cell. And so what I try and work on is understanding how the immune system works from essentially within inside the cell, because there's all these molecular processes that then drive what happens at the macro scale in the body. And that's what I realized once I started in this area. Even though it's just working on one protein, that's what I love. So I'm going to go into those details. If at all something's not clear, feel free to please stop me. So yeah, I was really happy that Katerina asked me to present this paper. So we published this towards the end of last year, and it was from my first PhD student who's finished now, and her name's Huijing Lim, and you can see on the first slide there that that's her photo. So she was from Malaysia and joined us in the lab and did her PhD, and this is the paper from her thesis, and now she's actually a postdoc at Yale, which I was really happy for her to get that position. And so all right, I'll explain what's happening in this paper. So the title is A Specialized Tyrosine-Based Endocytosis Signal in MR1 Controls Antigen Presentation to MAIT Cells. And there's the details there. I think you can probably get the paper. But actually, I'll just let you know, my email address is there, and I'm happy to chat to anyone if you wanted to contact later. And I'm also on Twitter there. Okay, so broadly, as you may know, the immune system is very complicated, and there's a lot of players involved. And without going into all the details, I've got a diagram of the main sort of cell types that are in the mammalian immune system, but this is even a simplification, really. And so we have like these two halves of the immune system. One is the innate system, and these are cells that respond really quickly to any sort of threat or pathogen that might break the barrier and get into your body. And they have they're termed non-specific because they don't really respond to just one strain. They often respond to a whole class pathogens, such as, you know, gram-positive bacteria. Certain molecules from all of those will stimulate those cells. And then there's, if the pathogen can get past the innate system, then the adaptive system kicks in. And that is very specific and can be much more powerful to tackle that infection. Although the adaptive response takes, you know, up to a week to sort of really get started. And so in the middle of these two, there's now these other cells that have been, you know, in the past couple of decades recognized as being important, and they're called these innate-like lymphocytes, or innate-like T cells. And so these are gamma delta T cells, NKT cells, and also this other subset called MAIT cells, and that's what I'm mostly interested in. And so there's a process that adaptive cells, such as the T cells, which are the main part of the adaptive response, or one major part, need, and that's called antigen presentation. And so whereas the innate cells can detect these molecules directly, adaptive cells actually need the antigen or these foreign molecules to be presented to them in a certain context by other cells. So that's called antigen presentation. So you can imagine one cell. I'll go to the next slide now. So on slide 3, the next slide shows that, you know, T cells, to get activated, need to have the antigens presented. So the blue cell is what we call an antigen presenting cell. And at that synapse, there's an MHC molecule, and it has a small fragment of a pathogen or a foreign organism within that. That's that little green blob. And the T cell has a matching T cell receptor that detects both the MHC molecule and that foreign molecule there. So you can see the situation's quite complicated already, but this allows for a really fine-tuned instruction to the powerful T cells in order to, you know, when to respond, how to respond, and in what way, because the antigen presenting cell can provide other signals in the form of like soluble molecules called cytokines and other receptors that can tell the T cell to either, you know, respond to this foreign antigen or not respond, and what type of response to happen. So our lab, and particularly my lab head, Jose Villadangos, has been working on antigen presentation for a long time. And when I joined, no one really knew how this new MHC molecule called MR1, it was newly recognized, how that worked. So I'm going to explain to you one important part of how it works today. So on the next slide, so slide 4, I'm going to introduce these MAIT cells. So MAIT cells are your most likely, as far as we know, your most abundant T cell that's in your body, and they're really powerful. They have very strong immune functions. And so the name MAIT stands for mucosal-associated invariant T cells. And it's because they were first found mostly enriched in the mucosal tissues, so in the gut and the lungs. But they've actually since been found that they're pretty much everywhere in your body, even in your brain. And they're extremely numerous. And so the invariant part refers to their T cell receptor doesn't really change much. And that's really different to most of the T cells in your body, because we all have potentially billions of types of T cells that can respond to billions of types of different antigens. But MAIT cells are very different in that they have the same type of T cell receptor that recognizes the same type of antigen presented by the same molecule, MR1. And so we think it's really important to understand how this happens. So what do MAIT cells do? Well, they can sense pathogens because MR1 presents these small molecules that come from bacteria, and that results in immunity. So they can, so for example, humans or mice that lack MAIT cells generally do worse for a lot of bacterial infections. And there's some humans that have had mutations that result in them not having MAIT cells, and they also often have some recurrent infections. But the other thing that MAIT cells do, which is really an interesting and emerging area, is that they they sense commensals and respond accordingly. So it's not just pathogens, because the antigen they recognize comes from all different types of bacteria. So they sense commensals, and then they can help repair the body. So in this diagram, this is like a mucosal layer, and you can imagine there might be a breach in that barrier where all these little green bacteria may be able to enter the tissue. So an antigen presenting cell can capture some of the antigen and present it on MR1 and turn on the MAIT cells, effectively turning on the immune system, because these innate cells, MAIT cells, can respond really quickly, unlike other T cells that take a long time to get going. They respond really quickly, and they actually recruit different T cells, and they cause dendritic cells to mature, and it's a real it's like it's really kick-starting the immune system and immune response. That's what they do. So we are very interested in what controls their activation. How does MR1 work? So a bit more about MR1, without going into these chemical structures in too much detail. MR1 is a really fascinating job, because what it does is it actually presents these small vitamin-related metabolites that come from bacteria. So I'm on slide 5 now, and so inside bacteria, most bacteria synthesize their own riboflavin, and as an essential vitamin, and it's vitamin B2. And we don't actually synthesize it, we get it all from our diet and also from our commensal bacteria that synthesize it. And so for organisms like bacteria and yeast as well that make riboflavin, if you've done any biochemistry, you know that to synthesize a complex molecule, there's a number of synthetic steps along the way. And one of the small intermediate steps is this molecule called 5-ARU, and that combines with these other small molecules to form the actual antigen. For example, the main one is called 5-OPRU here. So MR1 captures that small molecule within this little, what's called an antigen binding cleft, and then it presents it to the T cell receptor. So no other MHC molecule presents the metabolites like this. They all present peptides, as you may be aware, to conventional T cells, like the killer CD8 T cells, or some of them present lipids. But this is the first molecule that's ever found that captures bacterial metabolites and presents it to T cells. And the significance of this is that these metabolites are very short-lived, because they're very susceptible to being degraded. So if MR1 finds these metabolites around, it suggests that there is a microbe and it's living and it's synthesizing riboflavin, so it's essentially growing. So MR1 is sort of presenting this metabolite signature of a living bacteria. And these are really ubiquitous. Pretty much every bacteria we have, or probably, I'm not sure what the number, but it's more than, say, 90% synthesize riboflavin. So it's a very common antigen. So because MR1 is actually expressed by pretty much all of our cells, and the antigen is very abundant, so why are MAIT cells not continuously activated? And so we're trying to understand how MR1 works in those antigen-presenting cells, and I'm on slide 6, sorry, and in order to maybe find a way that we can manipulate MAIT cells, which are, to remind you, are your most abundant T cell, perhaps in immunocompromised individuals or elderly, where their immune response is not what it once was, and they're susceptible to bacterial infections such as pneumonia, we could boost their immune response. And there's also the scope for cancer immunotherapy, because maybe we could turn these abundant T cells to fight tumours, and there's some really exciting work that's been come out about that. So I'll explain what we found about MR1 briefly, because it has a lot of really fascinating elements to it. And one that I think was the most interesting that we found early on was that MR1, as you imagine, needs to get to the cell surface to present its antigen in order for the T cell to bind and recognize that as a receptor. But in fact, what people knew for a long time was that it wasn't at the cell surface at all. It was pretty much all buried inside the cell. So how does it actually get to the surface? So what we found is that normally in the healthy steady state, all MR1 is within the cell, so it's sort of it's off. It can't signal to the mate cells. But during an infection, these bacteria will release the metabolites, and they enter the cell by unknown mechanisms. But they get inside, deep inside the cell, in an area called the endoplasmic reticulum, or the ER. So on slide 7, you can see the bottom box shows that these metabolites are getting in, maybe from an intracellular bacteria or from outside the cell, and they load onto MR1. And so MR1's sitting there waiting just in case there's a metabolite coming around. Once it does bind, that causes a conformational change, like a change in the structure of MR1, and that rapidly traffics it to the cell surface. So the metabolites turn MR1 on, essentially, and MR1 is like an alarm system that signals to the mate cells that there is a microbe living nearby. And so once it's been at the cell surface, it needs to be turned off. And so the next slide, slide 8, I'm just highlighting this part of the pathway that we realized over a few years ago that MR1, once it's at the cell surface, when you expose cells in the top graph on the right, you expose cells to the metabolite 5-OPRU and MR1 goes up. And then if you take away 5-OPRU over the next 8 hours, MR1 goes back down. But no one knew how that worked. And that to us was really important because it's the way that you turn off the signal to stimulate mate cells. And it's not just that it goes inside to hide, but it actually goes inside and gets destroyed. So that's what that Western blot shows at the bottom, where you've got MR1, which is the black blob, and you can see that gradually disappears over time. So we wanted to figure out how that was happening. So this is where Huijing Lim's PhD project came in. So she found that MR1, we have this assay to be able to measure how fast MR1 is internalized. And that on slide 9, that's the top graph there on the left. And so with primary T cells, so primary PBMCs from human blood, different lymphocytes and monocytes, you can see that MR1 is internalized constantly over 4 hours, but at different rates depending on the cell type. So in monocytes, it gets internalized quite rapidly, whereas in B cells, it gets internalized quite slowly. And the same thing, similar happens in the cell line, 2 different cell lines, the THP1 and C1R cell lines. So to start to figure out how it is being internalized, we tried different inhibitors of endocytosis. So endocytosis is the mechanism by which surface proteins get internalized. And we found that with our 2 cell lines, if we use this inhibitor called pitstop2, which inhibits clathrin-mediated endocytosis, which is a major pathway for proteins getting internalized, that really stopped the amount of MR1 that gets internalized. So we realized that it must be some kind of clathrin-mediated endocytosis. And so on slide 10, this is where we used a very cool technique called genome-wide CRISPR-Cas9 screening to try and figure out exactly which gene was responsible for internalizing MR1. And these I'm not sure if you're aware of this. I mean, I'm sure you've heard of CRISPR, but the genome-wide screening is such a powerful tool that, I mean, it was a bit of a long shot, but we thought we'd give it a go. And we've done a few screens now, and they generally work quite well. So just to explain what happens, it's a way to knock out every single gene in the genome and then to see in a separate cell. So you have a large pool of cells and every gene gets knocked out in a different cell. And then you can sort out the cells that have a certain phenotype or a certain phenotype that you're looking for. So for us, we could see the MR1 slowly went down over time. And so we thought, we'll knock out every gene in the pool of cells, and then we'll isolate the cells that still have high MR1, so they don't behave normally, because normal cells will drop off MR1 over time. but we'll isolate the cells in that pool that can't internalize MR1 anymore. And then we'll find out what the knockout is in that cell, because that should be the protein coming from the gene that was knocked out is responsible for probably internalizing or degrading MR1. So when we did that, we found one hit, which is normally with these screens you find a few hits, but that can also mean that it's a bit confusing to know which one is the one that is actually mediating the effect. But we found this one really strong hit. It's called AP2A1, and this gene was enriched in all of our almost all of our repeats, our technical repeats. And with these screens, you have 6 different guide RNAs, so you can knock out the gene in potentially 6 different ways. And almost all of those as well, it was enriched in the MR1 high cells. So that suggests that that particular knockout is stopping MR1 being internalized. So we were quite lucky to get that, but then I'm not sure if it's luck. I just think that these screens are amazing. So I'll just explain to you what AP2A1 is. So it's part of a protein complex called AP2, and it's a clathrin adaptor complex. So automatically we thought, okay, this has got to be it. And if you look, so this is slide 11. If you look on the diagram on the right, you've got the plasma membrane there in grey, and there's the receptors in black on the cell surface, and you could just say that's like MR1. And what AP2 does is bind to part of the intracellular domain, and then clathrin binds to AP2, and then that enables essentially the recruitment of that receptor into these vesicles that form. And so these vesicles then are endocytosed into the cell, and then the clathrin and AP2 get removed from that. And then that's just a way that AP2 selectively captures proteins to be internalized and endocytosed. So there's 4 different subunits of AP2, and one of them is called AP2A1. So it seemed from our screen that when you knocked out AP2A1, MR1 wasn't internalized efficiently anymore. So we thought we were onto something pretty interesting. So we knocked out AP2A1 in 2 different ways in our cell line that we often use. So just to show you on the top left, here's our 2 knockouts. They have AP2A1 is in the 2 control wild type cell lines, but you can't see the protein anymore in the 2 knockouts. And then we use these to study how these knockouts affected MR1. And so on the top graph on the right, this is slide 12, you can see that that in our knockout cell lines, which is the blue line, the MR1 goes up at a much faster rate and then is slowly internalized. So these cells have a higher MR1 surface expression, and it suggests that it's not being internalized and degraded. And so when we measured the internalization rate at the bottom left graph, you can see again, the AP2 knockout cells have a much lower rate of internalization than wild type cells. And there's another protein called transferrin receptor, which is all of our cells have that, and that is what helps our cells take in iron. And the transferrin receptor, which is that's a known cargo for AP2. So AP2 binds to the cytoplasmic domain of transferrin receptor and internalizes it. That's like a control. So we could say with our knockouts that it was also affecting internalization of transferrin receptor. We also found that was quite interesting. There's been a bit of debate in the literature that because once MR1 gets internalized, a little bit can be recycled, which means that once it's been pulled inside the cell, it can also be sent back out. While other proteins like transferrin receptor get recycled to a really high degree, MR1, there's only a tiny bit that comes back out. And there was always a bit of a debate in the literature, just whether that is actually important for MR1 signaling to MAIT cells or not. And I'll get into that a little bit later. However, we found that when you knock out AP2A1, you also decrease the recycling to like by about 70%. So we wondered if MR1, this suggests that MR1 probably is a cargo that AP2 binds to, and AP2 is there to sort of turn off MR1 signals. So we thought this is, the next part of the MR1 story that we'd like to probe. And so it's really well known what type of proteins that AP2 binds to. And the first, on slide 13, the first type of protein that it binds to is proteins that have this motif called a tyrosine-based motif, which is the Y for tyrosine, XX for any amino acids, and then a phi residue. And I'll get into that in the next slide. And there's other types of motifs, which is in yellow, that binds to. But MR1 did have something that looked a bit like that tyrosine-based motif. So on slide 14, what we did was we looked to see if there was any of these tyrosine-based motifs, and what we found was that pretty much it was 80% of all mammalians' MR1 sequences have a conserved tyrosine residue. And they also have a conserved proline, but at that third position after the tyrosine, they don't really have what would be a canonical phi residue, which is that Greek phi symbol, because that phi is usually one of these 5 hydrophobic amino acids. So valine, isoleucine, leucine, methionine, phenylalanine. So normally one of these residues would be found in that phi position, but MR1 has this really conserved threonine residue. So we thought that was a bit unusual. So, but we considered that it could have a tyrosine that's needed to bind to the AP2 protein, but it lacks that phi residue, so perhaps it's sort of like a non-canonical motif. So we wanted to investigate that. So as I said, that the tyrosine-based motif in MR1, while being a little bit unusual, but it was very highly conserved. So look, I'm just showing you some interesting animals, as well as our standard Australian marsupials, like the wombat. But it's also conserved in the donkey and the blue whale. So just to give you an idea of how conserved it is, and MR1 is a very highly conserved protein as well. So we went ahead and made some mutants of these residues in that motif to see which one is important for internalization. And here's a bit of a schematic. So I'm on slide 16 on a schematic of the MR1 tails, and you can see that. So we made we firstly concentrated on the residues around that motif. So we made a mutant called Y313A. So we converted the tyrosine to an alanine. And then we also converted the tyrosine to a phenylalanine, which is F. And I've shown the side chain structures there. So you can see the phenylalanine is very similar to the tyrosine motif, but the alanine, basically, it lacks that aromatic ring. And then we made a series of other mutants, including cutting off the tail completely. And the very last mutant we made was to add a GFP protein onto the end. Because we already had that, we actually used that to image MR1 by microscopy. So just looking at MR1 internalization and the graph on the top right, there was only really one, aside from GFP, it was that Y313A mutant. So when we made that mutant, it significantly decreased the amount of MR1 that was internalized. So it suggests that that's really important for interacting with AP2, and we hypothesized that's probably the case anyway. And so also at the very end of the graph, when we add the GFP, that seems to interfere as well, and that's probably blocking the proteins from binding to the MR1 tail. So just to show you on the bottom, if you remember earlier, I showed that when we knocked out AP2A1, that reduced the internalization of MR1. And on the right, you see a very similar thing when we just make that single mutation in MR1. So we convert that tyrosine to an alanine. So it seems like MR1 has a very conserved tyrosine. It's essential for internalization, and that's probably dependent on binding to AP2. So we tried many different ways to see if MR1 and AP2 actually interact. And if you work at all in cell biology or biochemistry, you know this is something very standard, and reviewers will always ask for it. If you suggest that two proteins interact, then they want to see it. So it turns out it's very hard to do because AP2 interaction with other proteins is a very quick and like transient process. But we ended up using a method which is a microscopy method. So I'm on slide 17 now, sorry. And this is called the proximity ligation assay. And so the way this works is that when two proteins, if you look at the little diagram, the cartoon on the top left, if MR1 and AP2 proteins are in very close proximity within the cell, then the two antibodies against each of those proteins will be very close together. And then you get the proximity ligation assay spot, which is the spot of fluorescence occurring. So if you look at the black or dark microscopy images, at the top row, each of those little white spots inside is a point of interaction between the two proteins. And so we tried different conditions, and when we used HeLa cells that overexpressed MR1, we didn't see many spots, but only we saw more spots when we added the MR1 ligand, which is in the very last column of both the graph and the microscopy images. So I realized I didn't really mark that very well. And so it suggested that MR1 and AP2 were interacting just when the cells were exposed to the ligand, the metabolite. And we used PLA with two different types of antibodies at different subunits of AP2, and the same thing happened. So it suggested that MR1 doesn't actually interact with AP2 normally, but when that metabolite sends MR1 to the plasma membrane, the cell surface, then you actually do see the interaction with AP2, and that makes sense because AP2 is only active at the plasma membrane. That seemed to suggest to us that it is interacting. And we then were able to show that when you mutate that tyrosine, the amounts of PLA spots also reduces significantly. So that's looking at the grey compared to the purple dots there. So again, that suggests that even though in that tyrosine mutant there's actually more MR1 present in the plasma membrane, but when you don't have that tyrosine, you don't get the interaction with the AP2 complex. So we were really able to show that MR1 interacts with AP2, and it's very dependent on having that one tiny amino acid, that tyrosine. Okay, so we found, maybe I won't go into this too much detail, but we found that MR1, that cells that lack AP2 have, not only do they have more MR1 surface expression, but they present antigen for a longer time using these chemically modified MR1 metabolites. And so on slide 20, we found that, yeah, the same thing occurred when we used cells that were the antigen presenting cell in blue, and we could stimulate this MAIT cell. It's like a MAIT cell line. And when we knocked out AP2A1, those cells sort of had unregulated stimulation. So for a longer time, when they were just exposed to the metabolite antigen for a short time, they would continue to present MR1 antigen to stimulate the T cells. So this suggests that having AP2, in the cell is important in order to turn off the stimulation of that really abundant T cell. And so I alluded to the point earlier on that some labs had considered that the recycling of MR1. So I've highlighted, so on slide 21, I've highlighted in red here part of the schematic where MR1 is at the cell surface can be internalized, but then it can be pushed back outside. So that's called recycling. And some labs think that that's important because it could be internalized to a part of the cell where some bacteria might be living. And that does make a lot of sense because the bacteria are going to make the antigen, so maybe MR1 can head there capture some antigen and then return to the cell surface. But no one really knew whether was that really important for MR1 presenting or not, because we'd previously found that most of the antigens bind to MR1 when it's inside the endoplasmic reticulum. So, but the very difficult aspects to separate, because usually when you affect one thing in the cell, you affect everything else as well. So this is one of the caveats of doing sort of cell biology assays. So, but we found this was actually the unique opportunity, because when we knocked out AP2A1, the recycling of MR1 was reduced by around 70%. So even though MR1 was still able to load antigen in the ER and head to the surface and present, those molecules weren't really recycling very efficiently. And we think this is because AP2 isn't there to send it to the right location. So with this model in mind, we thought, well, now we can actually test, is recycling important or not? So what we did is we infected our antigen presenting cell with 3 different types of intracellular bacteria. And so we used Salmonella, Shigella, and Burkholderia. So this is on slide 22. And this graph shows the amount of activation that we saw in those MAIT cells. And we used wild type cells, which is the white bar, the controls. The AP2A1 knockouts in blue and the MR1 knockouts in red. So basically the MR1 knockouts don't really stimulate the MAIT cells. Those cells don't get stimulated. Whereas controls and the AP2A1 knockouts were equal at their ability to stimulate MAIT cells. So this really strongly suggested that if cells can't recycle MR1, it didn't really matter for being able to present the intracellular antigen. So it meant that recycling is probably dispensable. It's not important for being able to stimulate MAIT cells. That's a bit of an aside. It doesn't really explain a lot about that. It's more just an interesting part of MR1 and sort of settled a dispute for a long time, in my mind anyway. All right, so sort of getting towards the end, we wanted to figure out, why does MR1 in its motif have this this threonine in the position that other proteins have a valine, often a valine, or this phi residue? Because threonine is not hydrophobic at all, but the valine and all these other phi residues are these hydrophobic residues. So what we decided to do was to simply mutate that threonine to become a valine and then see what happened. And we also did a similar mutation. Well, we swapped the tail of another MHC protein called CD1D that has a canonical motif, and then we just swapped that tail onto MR1 to see what happened as well. But essentially, they both have the valine in the position that you would say an AP2 protein is recognized by. So on slide 24, we can sort of just go through this data pretty quickly, but in A, when we made these 2 mutations, you can see that MR1 is now is internalized much more rapidly. And it's also recycled much faster. So this is really opposite to what we saw when we mutated the tyrosine. When we mutated the tyrosine, MR1 was internalized and recycled much lower. But now when we make the motif look like a canonical AP2 sequence, now the internalization and recycling is gone up really high. And it was interesting that even though these mutant MR1s were recycled much faster, but that didn't help at all for MR1 presentation. In fact, the presentation was decreased. But this is also the opposite to what we saw with the tyrosine mutant. And on slide 25, we did a really classic biochemistry experiment that it's perhaps sort of fallen out of favor because it uses radioactivity, radioactive amino acids. So you have to be careful, wear double gloves and all that. But it's a way to be able to measure how long proteins survive in the cell. And there's really no perfect substitute for it, but so we still use this technique. And what we found was that you don't have to know too much about how this works, but on the top plot in the top square, you can see the MR1 at the end, at the very last lane of this gel. There's a bit of MR1 left. From 6 hours to 24, it does drop off, so it's being degraded a little bit. In the second box, the tyrosine mutant Y303A is really prolonged, so you see a lot of more MR1 at that 24-hour time point. Whereas if we mutate the threonine, it's pretty much all disappeared. So this shows that not only is it MR1 with these mutants, it's internalization being modified, but it's also how long it survives for. So we think these two processes are interconnected. So going on to slide 26, so just to summarize, what we think is going on is that MR1 seems to have this really Goldilocks motif, where it's not too hot and not too cold. So the cold version is the tyrosine mutant. So that tyrosine is really enabling the interaction with AP2 and essential for that. So if you mutate that, you get reduced endocytosis and it gets degraded really slowly. But then MR1 doesn't have that 5-valine residue. So if you put a valine residue there, then suddenly endocytosis is increased really rapidly and degraded very fast. So MR1, we think, has evolved to have an interaction with AP2, but not too much interaction, just enough. That's why I was thinking of the Goldilocks porridge. So just to summarize, so we think that this interaction is really governing what's happening to MAIT cells, and we think this is how MAIT cells are not always activated, and they're not always constantly secreting inflammatory cytokines, because MR1 is controlled itself, and that's in the residues in its tail and by what AP2 recognizes. So that's a bit of a whirlwind tour of one part of MR1, and perhaps you're not a cell biologist, but I hope you can sort of appreciate that all of our processes in our body, are governed by these molecular interactions, many of which we just don't know about yet. And so this is one, the MR1, which controls your most abundant T cell in your body. So there's a very last slide of acknowledgements, and I won't go through them all, but there's a lot of funding and people that I would thank, particularly my lab head, Jose, and Hui Jing, who did a lot of this work. And it was great to work with Hui Jing for my first PhD student, and she's now doctor. So yeah, thanks a lot. I think that's it. I'm happy to take any questions. Yeah.
Thank you so much, Hamish, and please tell from us congratulations and good luck for the future to your previous students. And yeah, this is really a beautiful work, and this mechanism, I think, or let me just ask, do you think, so first of all, do you think that, for example, in disease models, there would be maybe some sort of mechanisms that AP2 and MR1 interact too much to have kind of a chronic inflammation state? Do you think that
Yeah.
would be the case? Yeah.
Yeah, I think it's definitely possible. We don't really know much yet, and this is probably the limitations of how far we've gone so far. So we're still trying to figure it out now. But I did show you earlier on that some cell types can, in humans, like from human blood, some cells seem to internalize MR1 much more quickly than others. And so that suggests that there is some control over MR1 internalization, but we haven't really uncovered that yet. But one thing that's known is that proteins like MR1 on that tyrosine residue, that can be actually modified itself, like, say, phosphorylated, and that prevents AP2 from binding. So we haven't actually found that MR1 tyrosine is phosphorylated, but that could be, if that happens or something like that, any modification could be sort of, that occurs from another protein, could be modifying how much MR1 is internalized. But that could
Yeah,
certainly...
that's interesting.
Yeah.
Oh, no, please go ahead. Sorry.
No, that's it, yeah.
Yeah, I think it's so interesting because, when I read your paper, I kind of theorized like years ago that like diseases later on, like Parkinson and all these different diseases that come later on in some people, um, that maybe there is a tendency in those humans like to have an immune system that just doesn't stop or stops, um
Yeah,
doesn't really stop reacting. Like there's always a fire somewhere, a fire signal going on. And when I read this, I thought it would be really cool in the future. I mean, if we could detect, that's because this mechanism is on for too long, and maybe we could predict and like downregulate it in those people so they won't get Parkinson, or in the ideal world in the future. Your discovery
yeah,
would lead to, okay, we can predict this, so let's give them something that this
yeah,
mechanism is not all on all the time, and they won't get, I don't know, diabetes and stuff like that.
yeah. Exactly. I think you're right, and I mean I'm not sure whether this is involved, but there has been MR1 is really conserved within the human population and even between mammals, but there is some mutations in MR1 itself too, and I haven't looked yet to see whether there's any that are in the tail in this region that I was talking about, but that could also define, overreaction of MAIT cells or underreaction. But I think it's just important to know these processes because, how would we interpret, say, gene um, mutations without understanding how the mechanics work?
Yeah, thank you. And I'll pass the mic on to the next person. So in PTR order on my screen is I think Katie, Eli, Dr. Shah, Denise, Les. Just go ahead and mic, and thank you.
Yeah, thank you so much, Hamish, for your wonderful research. And I have two questions. My first question is about the using of the MR1 for the CAR T cell. And as you know that we are using for the monoclonal therapy and the cancer treatment for the CAR T. And another question that raised up after your presentation, because you mentioned about the different ligand related with the MR1, which it can be included folic acid and B12 relevancy. My question is, did you find any relationship between eosinophil and condition related with the eosinophil and MR1? This is two questions, and also about the APC. Do you think that the previous exposure to the viruses can be related with the expression of the MR1 or not? Thank you.
Yeah, thanks for the questions. Yeah, so the first question about like the CAR T cell, I'm not really up to date on this. I know that there is people working on actually like CAR MAIT cells. But I believe that's just because the good thing is that MAIT cells are conserved between humans and they can be transferred that way and expanded. Was there something specific about that you had in mind?
Yes, there is some research out there about the CAR T cell targeting mesothelia, and also they are using, because when we are talking about the T cells, specifically, no matter is a mucus related or not, they can have a use in a CAR T cell therapy.
Yeah, that's right. Yeah, so I'm not sure how MR1 um, uh, fits into it as well, because one of the things that MR1 is generally not at the cell surface to react. But yeah, look, I'm not sure if how far people have got with MAIT CAR cells, CAR MAITs, but I know that's
You mean
something people.
non-canonical signaling? You are mostly focusing on the canonical or non-canonical signaling for
Oh, for
this?
MR1.
Yes.
Yeah, with essentially just understanding, I guess, how MR1 signals to the MAIT cells. But by, I guess for therapeutics, we were sort of thinking that by understanding how it works and how it gets turned off, we might be able to interrupt that with, say, small molecules that block this interaction and then cause the T cell to become activated even more. Yeah. And so you're asking about eosinophils as well.
Yes, because you talk about the B2, and I was wondering about the folic acid ligand related to MR1 and how they are related, or you think they might be related based upon the cytokines activation or not.
Yeah, right. So, I mean, I know we think that eosinophils don't express MR1 at all, which is that's in mice anyway. That's a little bit, but the ligands themselves, there's some data coming out that MR1 can present these small MR1 these antigens from various different metabolite and metabolic pathways, but the main ones come from bacteria. But I haven't found any data yet that's about like eosinophils being involved specifically or any pathways from eosinophils. But it's a really unknown area because it was only discovered like 10 years ago that MR1 binds metabolites at all. So there's several labs around the world, and even there's a biotech company now looking at MR1 presenting antigens from tumor cells. But there's just a lot unknown. So any of the other innate like T cells could have their own type of metabolite pathways that enhance MR1 as well. But we just don't know that yet. That's an interesting question. So another question you asked about the antigen presenting cells and viral exposure and how that might impact MR1.
Yes, because
was that
specifically about the APC, and you mentioned about bacteria. My question was related to virus, and I was wondering maybe you have further explanation.
Yeah, I think that's really interesting because one of the things early on was that they, going back in the history, but they didn't know, the early scientists didn't know what MR1 was presenting, but they quickly realized that MAIT cells were never activated from a virus infection, but they were only activated from bacterial infections. So, and then later on, they figured out that it was, some of my colleagues actually figured out that it was these metabolites that only come from bacteria, so not from viruses. But I think what you're asking is really interesting because, obviously people that have a viral infection, they often get secondary infections later on and they can be bacterial. So we've wondered ourselves whether MR1 is, after a viral infection, there's some, immune training going on or within the APCs that's preventing MR1 from functioning properly. And we have actually found various inflammatory conditions in mice actually do decrease the MR1 presentation ability of these APCs, antigen presenting cells. I haven't actually looked at viral infections yet though. That's something we're interested to do. I guess this is what we're doing now. We're looking more at the in vivo role of MR1 antigen presentation and also what affects that. So this is exactly what we're looking at. But I'll have to do the viral infection and get back to you.
You mentioned exactly whatever I mean, and thank you so much for your answer. And I'll pass the mic to the next person.
No worries, thanks.
Yeah, Katie, Eli, Denise, Les, please go ahead, ask your question.
Hi, Amish. That was a great presentation. I really appreciate the detail. I had 3 questions. One, because I've never done this, how exactly is GFP, green fluorescent protein, added to a sample? I get that it starts with a vial, but then what?
Yeah, that's a good question. So what we do is we actually do some like cloning, like molecular cloning, where you sort of make up a chimeric gene sequence of, so you have MR1. Normally at the end of the MR1 gene there's a stop codon, so it means that the protein finishes. But what you can do is kind of cut and paste and put the GFP sequence directly after. So you take off the stop codon and the gene, and you have the sequence for the green fluorescent protein. Yeah, and then that means that in those cells, so then you ask that you get those cells to express that gene, and they'll make MR1 and just continue making GFP, and then it, you know, sort of miraculously folds correctly in the cell, and so you have the MR1 protein in the membrane with the GFP hanging off the end. So it's all one big protein.
So how long does that take, and is it like, what is the equipment involved? I'm just really curious about this.
Yeah, no, it's no worries. It's a good question because, you know, there's probably a lot of things that I've said that maybe didn't make sense because if you're in a very different area. But basically, the process might take like maybe like a month or something, or a few weeks to get going, because you have to do various techniques like PCR, so polymerase chain reaction, and they're very classic molecular biology techniques where you stitch the two genes together, and then you put it into a vector, which is like a plasmid, a circular piece of DNA, and then you can manipulate these viruses to inject that DNA into the cell that you're interested in, so a cell line that you're growing in culture, and then the gene will get inserted into the cell. So this is, you know, when they say standing on the shoulders of giants, like these techniques have countless scientists have done every step and optimized and made these work. You know, it's a mystery to me almost how it works, but we can do it. So yeah, it's pretty cool.
Thanks for that explanation. That definitely clears up some questions I had. You had mentioned, if I didn't hear you wrong, AP2 timing is fleeting, and that's why it's difficult to prove this. How fleeting is that?
Yeah, so let me see if I've got the. Basically, what happens is that endocytosis itself, which is a process of, you know, parts of the surface of the cell, the plasma membrane, forming these little pits, and then they get pinched off into a little bubble kind of thing, and then that gets taken inside the cell. So that's called endocytosis, and that happens in the seconds, like say 5 to 10 seconds. But what AP2 does is even faster than that. So it binds to the proteins and then sort of collects them together into these pits, because it's the clathrin protein that binds to AP2 makes it this scaffold. Think of like a golf ball forming with like these scaffolds around it that sort of make it into a pit, and then it becomes a ball. Another protein comes and pinches it off, so it's a ball that floats off from the membrane inside the cell. So that happens over seconds, and the AP2 interaction, I can't tell you how fast it is. It may be the data's out there, I'm just not sure, but I'd say it must be less than, you know, a couple of seconds. So it's sort of AP2's binding, collecting proteins together, sending them into these pits, and then once the pit becomes an actual like endosome, a ball inside the cell, the AP2 dissociates. It seems to happen very quickly.
Well, that would, yeah. I mean, the order of seconds, you have to be at the right place at the right time with the right equipment.
Yeah, exactly. That's right. Yeah.
And then the last question is, how have you considered how your findings for this work apply to COVID? Is there, I'm sure there is some application. I'm just curious if you've considered it.
Yeah, so we don't typically think of MR1 being really involved in viral infections because MR1 presents these bacterial metabolites. But there has been some work to show that, especially in mouse models of influenza, I know it's very different to COVID, and it's a viral infection, and MAIT cells are actually important part of the immune response to viruses, and that was a bit of a surprise because MR1 doesn't present any antigens from viruses. but it does show that MAIT cells, their role in the immune system is probably more more important in other aspects like viral infections that we just don't know yet. But the MAIT cells are so abundant that you can't really ignore them. So there's been data to show that MAIT cells are almost like a canary in the mining tunnels, where as soon as something starts to go wrong with the canary, there's a problem. And same with MAIT cells. So a lot of diseases, autoimmunity, diabetes, COVID, MAIT cells their numbers drop in these patients. So there's definitely something happening to MAIT cells. But going back to your question of, is what we found, how does that relate to COVID? The only thoughts that I've had is that I think, and this is going back to a previous question as well, is that perhaps after a viral infection, if MR1 itself is impacted and not able to stimulate these MAIT cells very well, which we think is probably the case from other data, then that means that people that have had COVID or a viral infection could be, at risk of getting lung pneumonia or some other viral infections, because the MR1 is not working properly, and MAIT cells themselves are also they get reduced in numbers. So it could be. So yes, I'm sure. I think everything does come back to COVID at the moment for us, but it could be something. And if we can manipulate MR1, it could be a way to also boost the numbers of MAIT cells back up, because as I said, they do drop it after COVID or during COVID, and they get activated. But with MR1, if we could, boost the MR1 presentation on our cells, maybe we could also return the MAIT cell numbers to a normal level quicker than what normally happens.
So I actually searched this question during your talk. I nearly went. There was a time I wanted to go into immunology, and I've still always followed it to some degree. So MAIT cells are new to me, so this is really interesting. But on this specific topic, there was a paper from 2021, Isabelle Nel at l'Université de Paris. I'll read the specific sentence that caught my attention on SARS-CoV-2. We and other groups recently unveiled alterations of MAIT cells during SARS-CoV-2 infection. Blood MAIT cells were significantly reduced in frequency, while a highly activated high CD69, CD56, interferon gamma, and GZB expression, and those parameters were linked with pulmonary function loss, global inflammation levels, and fatal outcome of severe patients. Elsewhere, here. MAIT cell frequency was increased in infected lungs and also displayed higher levels of CD69, GZB, and PD1 compared to blood MAIT cells. Now, PD1, program death receptor protein, that is associated, elevation of that is associated with long COVID.
Yeah. Yeah, that's right. So basically, what's happening, what they're describing is that MAIT cells are decreasing in the blood, but they're going to the lung because that's the site of inflammation, infection, and so they're becoming activated. So they're expressing all those markers. I think that's that's really interesting. They're probably MAIT cells are being taken out of the blood, going to the lung, and potentially causing further damage. Although it's I'm not sure if it's like causative or correlation, because MAIT cells, like I said, a bit of a canary. They get activated very quickly. They can often be activated even without MR1, so that's probably what's happening. So even just some inflammation that's causing cytokines to be produced can cause MAIT cells to become activated, and they secrete these inflammatory cytokines like interferon gamma that you mentioned, and that's causing potentially further damage. So
And
maybe in this case,
the title of sorry, go ahead.
I think if we were going to try and manipulate MR1, maybe we'd actually in the lungs try and turn it off in case MR1 is actually further exacerbating the this inflammatory MAIT cell response.
So the title of
Yeah.
the article is "MAIT Cells, Guardians of Skin and Mucosa?" ? They, like in the same paragraph that they talk about SARS-CoV-2, they also talk about influenza A virus, and there they note that MR1 double negative cells are associated with worse outcomes.
Okay. Yeah. I'm not sure what
So
that
I'll
is.
put the link to this paper in the room chat for you.
Yeah, thanks. Yeah, I do know that one. Yeah, it's a good review. Yeah, pop it in the chat. That'd be good. Thanks.
That was all the questions I had. Who else would like to ask questions?
So actually, I
Thanks.
did have a question. You had the slide with the various bacterial pathogens. Do you know if like during natural bacterial infections they wind up inside mate cells, and are perhaps mate cells kind of a trap?
Yeah, good question. I'm not aware of studies that have found them residing in mate cells, but they certainly could. I mean, a lot of bacterial pathogens that want to get inside cells, they usually get into either phagocytes, like macrophages, because they're constantly eating bacteria anyway, and say like tuberculosis and some pathogens like to get in there, and they end up making their macrophage their home. I'm not sure about T cells though, although look, I'm sure there is a pathogen that gets in, but a mate cell is like just another T cell, but it does have important differences. But I'm sure that there's some bacteria, we haven't found any, but I'm sure there's some that can manipulate mate cells. So maybe they increase the activation, or they decrease, and they don't seem to be bothered by them. But for example, like tuberculosis, the bacteria that causes that, mycobacteria, that does seem to be susceptible to mate cell activity. So the MR1 does present antigen from those and can stimulate mate cells, and there's various human data to show that's important in the immune response, and mouse data as well.
And also, I'm wondering, since you brought up the difference between commensal and pathogenic bacteria, is there a difference between commensal and pathogenic bacteria in terms of? I'm thinking of like the soil microbiome where desirable organisms get stuff from plants in exchange for the things they do for plants, and perhaps whether they are getting the B vitamins so that they don't metabolize them on their own, and maybe that's a way that they don't induce a response because they have less of the metabolites.
Yeah, so it's something that really fascinates me, and it's something that I'd love to understand. And I guess one important thing to think about is that with these T cells, including mate cells, as I just said at the beginning, is that they need to be presented, the antigen, in by another cell with a protein like MR1. So that allows this a context or another story to be told at the same time. So it has this really good layer of regulation. So why that's important is because some labs, and I'm thinking there's Yasmin Belkade at her lab. I know that she's shown a lot that the microbiota can influence immune cells, but it's not what people normally think of in immunity during inflammation. It's like in the absence of inflammation. So antigen presentation can occur, and it does certain things to the immune response and the tissues that isn't really a bad thing. So, for example, what I think is important is that commensal bacteria, I don't think it's a bad thing that they have these antigens, because if there's no inflammation in that tissue, say on the skin, then what happens is the MR1 probably presents their antigens, but there's no other inflammatory cytokines or signals going on. So the MAIT cells probably just say, okay, I'm detecting some MR1 here. I'm just going to proliferate a little bit. I'm just going to sit in these tissues just in case something happens in the future. So I don't think there's really probably a difference, as far as I know, between commensals and pathogenic bacteria in their antigens they produce, like the MR1 antigens, the metabolites. It's just the context that happens. So probably things like a lot of tissue damage, or maybe excessive amounts of the antigen because pathogenic bacteria are trying to multiply much more rapidly. Maybe these signals suddenly swing from a sort of normal, healthy antigen presentation, MAIT cell activation response to something that now MAIT cells are thinking, okay, this is actually starting to look bad. Now we'll start to not just proliferate, but we're actually going to start secreting cytokines and kickstart the immune response. Does that sort of answer your question?
Yeah, very much so. Thanks.
Yeah, no worries. I think this is what I really find fascinating. You know, we don't know yet, but there's all these other clues from other fields of science sort of suggesting that it's all about the context from which the antigens are presented.
Yeah, thank you so much. Did anyone has any more questions? I think Les he left. But let me check the chat so far. So far I think in the chat, please raise your hand if you have any more questions. And yeah, if you have follow-up questions, anyone, please go ahead. So what is the next step you're doing in the lab now, or will do when you go back? What are you planning to do next in this?
Yeah. So we're looking a little bit further. I've sort of got two main areas that we're pursuing in our team. So one is continuing this sort of cell biology, because we think there's more to the story of the degradation of MR1, like how once it's internalized, we know that it's degraded, and how that's happening. Is that specific or not? And also MR1, the antigens for MR1 get inside the cell somehow, and we don't really know. That's not well described. So we're trying to figure that out now. The pathway that these antigens, metabolites, get imported into cells in order to send them onto MR1. That's actually a very unusual process. The MR1 is deep inside the cell, but it's actually sensing something from outside the cell, and so we don't know how that happens. And so that's something we're pursuing as well. However, the other part that I'm looking at is it's a very basic question, but which cells actually express the most MR1 in the body, and is there some cells that are specialized in presenting and making MR1 and presenting it? So we're looking at that in mouse models at the moment, and what do some previous infections do to MR1? Does that prevent MR1 presentation or not? And we have found that if mice that have had a bacterial infection, at least we've looked at now up to 9 days after that, and their MR1 expression in the main cells that express it is turned off almost completely. And that was really unexpected, because we thought that, during an infection, maybe MR1 would be turned up, because it has a very low expression anyway. So, but it seems like after an infection, MR1 is turned off completely, and we're not sure when it recovers yet. So that's what we're looking at. So we'd like to see if the same thing happens in humans. And, so I've had some great questions today, because it is what we're sort of pursuing, what's happening after a viral infection, or a primary bacterial infection. Does that lead to, is the MR1 reduction, is that leading to increased secondary infections? And that's a possibility. So without giving too much away, that's what I think is happening.
That sounds really interesting, and yeah, I'm really curious to learn. I can't wait until you learn more about that because, yeah, as I said, I think it has so many implications how this mechanism works in like maybe chronic disease also.
Yeah.
Although you say it's relative specific to bacterial infection, but maybe it could give us a hint how the immune system in that person in general, like if it's overreactive,
There,
Yeah.
basically. And
there were a
I
couple of
mean,
other papers as I was searching that talked about mate cells being reduced after COVID.
right. Yeah, yeah. And that's just probably the blood because it's the blood's obviously easier to get from humans, relatively. It's not very invasive. So that's the thing, like I was saying, it's like a canary because the mate cells always seem to go down. Every single disease, they seem to go down in the blood, but whether they increase somewhere else is often not known. But yeah, like you said before, they do increase in the lung during COVID. Yeah.
That's always an interesting question, right? I mean, there was X quantity, now there's Y quantity. Where'd the rest go?
Yeah, exactly. And surprisingly, it's not that easy to say if they've died or proliferated or just gone somewhere else
Well,
in
the,
humans, at least.
the fact that they're expressing PD1 suggests that apoptosis would be something to look for.
Yeah, that's true. Yeah, yeah, that's true. Yeah, it suggests they're exhausted and maybe about to die. Yeah.
so does the training, um, the training of the system also for this made cells, does it occur during development or are they like how does it work? I'm sorry that I don't know that, but
Oh, yeah.
is it
No.
yeah.
Oh, I think the short answer is probably not really well known, but um, it's just emerging all the time, like what mate cells are doing. So there was a paper this week in Immunity showing how they help repair like wounds in the skin. And I sort of mentioned that before. That's been known for a little while, but they've just gone a bit further. There was a paper like a month ago in Nature Immunology showing that mate cells are involved in maintaining the meninges around the brain, which was really like sort of unexpected. Um, but so who knows like what they're playing in development. I don't think there's much that's known, but I'll give you a brief idea is that when when babies are born, they have there's not that many mate cells, but they over the next few years of life they start to expand and become the most abundant T cells. So by the time I think it's around the age of 30, which I'm past, well past, mate cells are at their peak and then they start to decline again later on. So mate cells could certainly be playing a role, but I can tell you that they expand a lot from birth, and then sort of mature adult, and they start to decline. But they could be having functions in the brain. They could be having functions in development. Um, who knows? It just hasn't been looked at.
So does the thymus play a role in this or not at all?
Yes,
Um,
the
um yeah.
yeah. So the thymus is where mate cells are developed. So mate cells are sort of they get trained in the thymus like other T cells, and they leave the thymus and then expand more in tissues. But MR1 is presented by certain thymocytes. They're called double positive thymocytes, and that instructs these developing mate cells to become mature mate cells that leave the thymus. And one of the really interesting things, actually, I think there was a paper. It was 2 years ago in Science from Olivier Lance's group in France. So he's the pioneer of mate cells early on. But he found that these metabolites that are from commensal bacteria in young mice they actually, from those barrier tissues, like say the skin or the gut, those metabolites somehow get transported to the thymus, and then they get presented by MR1, and that helps develop MAIT cells. And that was really unexpected because it's only been I think most immunologists consider the thymus a bit of a closed system where all the antigens come from within the cells in order to train T cells. But this is a case of where it's a foreign antigen being transported inside in order to train these MAIT cells. So yeah, I'm not sure if that sort of answers your question,
Yeah,
Katarina.
that's really interesting because I heard or read shortly, but it was a while, like a bunch of months ago, that um a medical researcher was trying to transplant thymus, and it kind of trained. It was not a big problem if you do it early enough during development, because I think she trained the thymus first in a dish and then transplanted it, and there was no rejection and things like that. Um,
Yeah,
Combined with like the immune system, for cancer treatment. I thought that was really interesting, and then I thought, but the thymus training goes on really early in life, but I thought that was interesting if you could basically also manipulate the thymus in order to, I don't know,
yeah,
then improve if you have like, but that doesn't
yeah.
work in adults. Let's say you have a long-term COVID and the immune
Mm,
system doesn't bounce back regularly. Could you train the thymus to train? But that's too
yeah,
late, right? That doesn't work anymore later on. So it was like a misconcept in my head. But that's interesting. Thank you.
yeah.
So
I don't know. I think I'm all open to other ideas. It could be something to pursue.
yeah, and then would the like epigenetics that maybe get inherited, kind of, if modulate this? Like, could that mechanism be modulated by those mechanisms? Is there any hint that that would be the case?
Yeah, not that I'm aware of, but I mean, I can imagine. I have seen that different human donors, like that I've looked at their MR1 expression, does vary, and so we don't know why. That could be epigenetics, or it could just be to do with the state of that person at that time. It could be diet, who knows? But yeah, it, it's all open for. I've got no idea. But even just the expression of MR1 seems to change.
Yeah, that's interesting because yeah, if it's like a early developmental thing, that it's highly dependent on really relatively early stages in life, then maybe, kind of environmental factors. Maybe there's like a risky stage where you should be careful to not, change that system, basically, if
Yeah,
the epigenome would be able to modulate it.
yeah, that's true. Yeah, that's true. Yeah, it'd be worth. Yeah, we need to know. I think we do need to know that. Yeah.
That's interesting. So is there like a large set of genetic data you could go through to see if, in different populations, I don't
Hmm.
know, genetics or
Yeah,
I don't know, methylation levels or so are changed?
yeah. I'm not really aware of it. I know. I know there's been some studies on looking at just the gene sequences of MR1 and how it changes. It's very well conserved, but there's a few differences. Someone found that this is not so this is obviously not epigenetics when it's at the nucleotide level, but there's like one single nucleotide difference in some people's MR1, and what they found is that it changes the expression of MR1. They think that it changes the expression.
Oh,
It's fairly
wow.
rare. Yeah, it doesn't change it a huge amount, but actually they found it in a cohort of Vietnamese people, and they had an increased susceptibility to tuberculosis. So it does suggest that MR1, MAIT cells are important in tuberculosis, but I think it was actually the mutation increased. They thought that it increased MR1 expression. I believe it was like an in silico prediction, so I'm not sure if it panned out to actually if they did analysis in the humans, in the people at all. But anyway, they found that increased MR1 increased the susceptibility to tuberculosis. So I'm not sure what's going on there.
Yeah, that's interesting. I thought it would be maybe, we had the guest speaker, it was a completely different context, but she looks for her work through data sets from hunter and gatherers to kind of make evolutionary analysis, basically, because, the assumption is that in hunter and gatherers that still live that lifestyle,
Yeah,
there may be something conserved from back in time, and then you know the diet is very different, more active lifestyle and so on. So it would be really, yeah, I would be curious how, if it would be different there, if they would be less susceptible to bacterial infections. And so I think that would be also interesting if there
yeah,
would be indefinitely amount of funding.
yeah.
Probably.
Well, I think that the MR1 is there to sort of monitor the commensals and the microbiome to some extent and instruct the immune response. That's what my thinking is. And in our the Western lifestyle, that's been changing the microbiome significantly. And so who knows like what MR1 and MAIT cells were like. I'd love to know. But there's it's clear that like mice are very clean. The mice that we use in the lab, they're called specific pathogen-free SPF mice. They don't have many pathogens. They also don't have many MAIT cells. It's hypothesized that that's if you boost the diversity of the microbiome, that maybe MAIT cells will increase. So maybe in humans, as we're altering our microbiome, our MAIT cells are not really being trained enough by the commensals. But we're not sure about that yet.
Yeah, and the microbiome diversity is also declining significantly in us, but also in our environment through, extensive farming and pollution and so on. So yeah, that's
Yeah.
another very interesting aspect of your work. So yeah, we've been talking for like an hour and 40 minutes. I wanted to, give back your time
That's
because
all right. I probably
you
will.
have
I probably
children.
will go. Yeah. I think my toddler had a bit of a tantrum before. He's in. I don't know if any of you heard this, but he's a three-nager at the moment. He's 3 years old, and he's so I hope that he's finished now. But I might need to go and see how my wife's going.
Yeah,
Um,
I,
but
I, yeah, I hope all is well.
Well,
no, I'm sure.
thank you for a fascinating talk.
No worries, my pleasure. Thanks for the great questions. It's been I love being able to talk to people from very different backgrounds and their different thinking. So thanks.
Hi, it's Katie. I just also wanted to say thank you, Hamish. You did an amazing job, really, breaking down some very complex terms and everything like that, communicating very well. Welcome to Clubhouse as well. I hope you come back and join us. And just really, really quickly, because I'm fascinated by your research, I also went down a rabbit hole during your talk. And if you do want any blood samples from people that have had COVID or have long COVID to test this, you please get in touch, because I can get you in touch with different labs around Australia studying this sort of stuff. But also, if we want to follow up with your research, you mentioned a Twitter account before, like how do we follow up with you and follow your work? I also posted an article from, I think, your university in the chat below as well. But if you can share your Twitter handle, would love to follow you there.
Yeah, no worries. I'll put it in the chat. I also put it on my slides. So I'll just put it in quickly. It's It's HMCW_science. That's my Twitter. I just put it on the chat. So yeah, I'd love anyone to get in touch on Twitter. And yeah, while Twitter's still around, it seems like it's hanging on for a bit. But yeah, no worries. Thanks so much.
So are you planning to go on to Science Mastodon? There are a couple of good ones.
I would like to. I it's been in the hard basket at the moment, but I was just waiting to see what happened. But it sounds like a lot of people are moving there. Yeah, I haven't signed up yet, but I need to check into that. Yeah.
I need to check
Yeah.
that out too. Thanks,
Yeah.
Eli. Okay,
All
thank you
right.
so much,
Thanks.
Hamish. Say thanks to your kids for, making, like, letting us share, like, sharing you with
Oh,
us.
no
And
worries. Yeah.
yeah, it was a
No,
wonderful discussion. Thank you so much.
no worries. Thanks so
And
much for the invitation, and yeah, I'll see you around in Clubhouse soon.
yeah,
And
and enjoy your summer while we suffer. No, I'm kidding. Enjoy. Okay.
no worries.
Thanks everyone for coming.
Thanks so
And
much.
yeah, and follow the club if you like discussions like this. And the next discussion will be with Dr. Benjamin. He was actually in the audience earlier listening, and he will talk about naturally emerging neural codes, which is really interesting, a really interesting neuroscience topic. So, yeah, I hope I hear you all back soon. And yeah, enjoy the rest of your day, Hamish, and everyone. And I'll close the room in 3, 2, 1. Bye, everyone.
Yeah.
Thank you.